Good afternoon, distinguished guests and valued partners. My name is Harry He. Welcome to Viva Biotech's Enchantment of Drug Discovery launch event. We're truly honored to have you here, both in person and online. We celebrate a new milestone in advancing innovation. AI has evolved from theory to impact, from the Nobel Prize to QM, MM, to breakthroughs like AlphaGo, AlphaFold, ChatGPT, and DeepSeek. AI is truly transforming how we solve complex challenges. In drug discovery, AI accelerates target identification, molecule screening, design, and beyond. By integrating computational insights with experimental testing, AI makes the drug discovery process faster, smarter, and more efficient. At Viva Biotech, we pioneered the conjunction of AI with structure-based drug discovery, creating an AI-empowered, structure-enabled platform to explore new target, new mechanism, and new modality. Through this innovation, we proudly deliver a one-stop, first-class discovery solution to our global partners. Now, it is my great pleasure to introduce Dr. Yue Qian, head of the AIDD CADD platform at Viva, who will share with you our vision of artificial intelligence in drug discovery and Viva's role in reshaping the entire drug discovery process. Now, let's welcome Dr. Qian.
Thank you, Harry. Thank you all for being here. It is my great honor to present to you our Enchantment of Drug Discovery today. Artificial intelligence for drug discovery to many people are mysterious and hard to understand, like a form of dark magic, powerful yet intimidating. It is true that this is truly complex. It's an interdisciplinary field of chemistry, physics, biology, computer science, math, and medicine. However, our mission here is to fully understand and explore AI's potential and leverage its power to resolve our real-world drug discovery problems. So let's take a look at the evolution of the artificial intelligence first. Conventional machine learning, or ML models, are trained on complicated patterns to learn these experimental data sets, with the most well-known as QSAR, or quantitative structure-activity relationship. It's still a very popular tool to this day. In the late 20th century, neural network emerged in exploring and predicting complicated patterns between chemical structures and observations, such as bioactivity and toxicity. Its applications widened as the data accumulates and the growing of computational power. Recently, large language models, or LLM, has made profound impacts in drug discovery, with the most well-known being ChatGPT. And protein LLMs are like ChatGPT for biology, where they learn from millions of sequences to be able to speak the protein semantics and enable downstream tasks, such as protein structure prediction. And this is what AlphaFold 2 does. AlphaFold 3 takes one step further by not only reading biological sequences, but also chemical structures, and output their interaction network in the form of 3D coordinates. So essentially, AlphaFold 3 is a mixture of biology or protein LLM plus chemistry LLM on the basis of structural biology. And we believe such combination is the key to its success. While AlphaFold 3 is most relevant in the beginning part of drug discovery, moving forward, we believe the same philosophy is crucial in fully exploring AI's potential. So that means the integration of structural biology, molecular biology, medicinal chemistry, biochemistry, and pharmacology. AI models being able to manage large data sets and extract hidden features and learn the intrinsic patterns and ultimately make predictions and generate novel ideas is how we envision that we will be able to decipher the mystery of new targets, new modalities, and new mechanisms. Viva Biotech, as a one-stop drug discovery platform, has already encompassed all of the aforementioned aspects. Our philosophy in revolutionizing drug discovery roots in the atomic understanding of the biological system. We've been built upon this principle for over a decade, backed by our very powerful and world-leading structural biology platform. We've established high-throughput affinity-based screening platform, along with a comprehensive list of biophysical and biochemical assays. We're very proud of our medicinal chemistry team and the biologics team. Our expertise expands to pharmacology and manufacturing. So these capabilities in the outer ring represent key factors that we input into our AI model. They are structural insights, assessment to affinities, late-stage drug candidate properties, as well as real-time wet lab feedback. Viva Artificial Intelligence in the center functions as the neurons, as the edges connecting the dots, or neurons to speak the language of deep neural network. So in this process, we're improving our data-driven and principle-driven models iteratively over the time. Empowered by these capabilities and our years of first-hand drug discovery experience, Viva Biotech developed our unique way of maximizing AI's power in a way where we integrate all of these key components. Let's take PROTAC as an example to further illustrate why it is so crucial to leverage both biology and chemistry and beyond in understanding the drug discovery process and fully leverage AI's power. Each of you here has been gifted with a 3D print miniature. That's a PROTAC in complex with a protein of interest and a E3 ligase. The ternary complex structure is first solved here at Viva. PROTACs are new modalities, and they represent new mechanisms by facilitating ubiquitin transfer and subsequent target degradation. Unlike affinity-driven binders, these are PPI-driven binders, meaning protein-protein interactions. So conventional cheminformatics tool would often fail because it couldn't capture the multi-component landscape in these scenarios. And vanilla protein-protein docking wouldn't work as well because proteins were treated as rigid in those tools. Acknowledging that there are protein-protein interactions playing a key role in these systems, we use AI-based scoring functions to score and rank these decoys using such as IPTM or GPDE. And recognizing that these proteins may experience significant conformational change, we use molecular dynamics to sample and refine a ternary complex structure to enable the follow-up degradation prediction and generative linker design. And that's how we fuel our mechanism-aware model with relevant data under the guidance of structure-based drug design. This is how we are reshaping the drug discovery workflow at Viva. For small molecules, traditionally it takes about 2 to 4 years to bring a drug candidate all the way to the clinical stage. And the sequential process of optimizing one property at a time is very inefficient in the sense that we need to go back and forth between these steps. In the AI-driven workflow, the entire timeline is reduced to well under 1 to 2 years. So in a sense, structural modeling coupled with de novo design and virtual screening would be the very indispensable step to initiate the project, followed by molecular generation and experimental validation. In this way, we're sending the drug candidates into the lead optimization stage in a much faster manner and with even fewer design, make, and test cycles. Throughout the entire process, we closely monitor all of the properties at the same time to increase its efficiency. Therefore, we can make such drug discovery process 2 to 3 times faster and effectively reduce 50% to 70% total cost up to the preclinical stage. We've adopted a brand new antibody discovery workflow as well at Viva. Instead of using conventional animal immunization or in vitro display methods, we take a completely different route, starting from AI-generated sequences. These steps takes only less than a week, with most of the time being the research scientists reading literatures before clicking that generating button. Because these sequences are rationally designed, we reduce the total number of sequences to up to 10 to the 7th order of magnitude compared with phage display. And that enables the lab validation to be able to finish within 2 to 3 weeks, providing valuable data points to our model refinement step. The entire cycle takes about 4 weeks, and by then we're expecting several promising, appealing antibody candidates. One of the advantages of the AI-driven workflow is that it takes account into the positive and negative data points as opposed to conventional screening methods. And the models are learning patterns that are generalizable to make it target independent. Therefore, in our way, we can increase the total efficiency by over 400%. And according to our own experience, it exhibits about over 85% success rate on nominating at least one candidate with desired properties. Today, for the first time, we would like to introduce Viva's web-based artificial intelligence for drug discovery platform. It comes with 3 modules that are named after 3 death hallows. V-Scepter represents the elder wand that empowers the computational modeling with fundamental rules. V-Orb reviews the underlying mechanism with principle-driven methods. And Mantle is the invisible cloak weaved at Viva to help us explore the hidden space of drug discovery under the guidance of generative AI. Specifically, V-Scepter includes pocket identification, force field parameterization, molecular docking, ADMET predictions, and a series of other useful tools. And V-Orb consists active learning augmented virtual screening, protein dynamics, enhanced sampling, and our proprietary free energy perturbation platform that includes both non-covalent and covalent, as well as biologics modalities. And V-Mantle encompasses protein large language models that works as foundation models to extract features and enable downstream tasks. De novo design and structure prediction work as the key pillars of the state-of-the-art artificial intelligence for drug discovery. And lastly, antibody engineering platform resembles a streamlined process where we use to design antibodies in the most accurate and efficient way. We will talk more about these highlighted tools in just a few minutes. In leading the era of AI-driven drug discovery, we believe it is important and really crucial to engage data, algorithm, mechanism, structure, and the integration of wet and dry lab. With that, let's move on to the next part. In the next part, I will work with my colleague Junhao to show you some of the features on our website. And first of all, I was wondering if any of you recognize the small molecule that unlocks the door to our lab space in that demo video. It's AZ0780. It's the allosteric inhibitor of PCSK9 that was first designed here at Viva. So the molecule itself is identified through the process of fragment screening and a series of hit-to-lead and lead optimization with the help of biophysical and biochemical assays, as well as a co-crystal confirmation of the complex structure. And the compound is currently in phase 2, the latest result released by AstraZeneca being that the patients are able to hit their target DLDC level, and that's giving us promising results that's supporting further development. So let's take a look at its ADME profile. We've measured some of the key values for this particular compound experimentally, such as KCO2, plasma binding, live microsome T-half and clearance, as well as CYP and hERG toxicity. And since we've implemented all of our ADME predictors into our website, it's fairly straightforward for us to take a look at all of these, the calculated values of these terms. So let's move on to the ADME predictor in here. We've built the interface where we can either draw the molecule directly or input a series of SMILES code, or upload an SDF file to calculate these properties in batches, or load the example, which happens to be this molecule. And if we draw or edit the molecules in the window, the SMILES in the bottom changes inherently. So these two goes together. And upon clicking the submit button, we're brought to the user center. And in here, we've already had several completed tasks for testing, and that can bring us to the calculated results. And this is a list of the full batch of files. And if we open one of them, or if we take a look at the AZ0780 directly, we would be able to see a 3D display of the molecule on the left panel and the physical chemical property on the right-hand side. Down there, the table lists the full property that's calculated with our model. And let's bring these data back to our table and see how well they align with each other. It's really intriguing to see that out of the 14 properties we calculated, 12 are within the error range. So that represents 80% of the accuracy in predicting the measured properties. Before moving on to more statistics, I want to share with you one of the unique approaches we take at Viva to build these models. Conventionally, ADME properties are usually treated with cheminformatics tools. So that means we only look at the chemical information and disregard the rest of the information. However, in our way of understanding it, we believe chemistry, biology, structure, modeling, algorithm, everything should all be put together. So taking the CYP stability model as an example, our way of understanding it is to not only look at its chemical representation, but also to put the substratal and inhibition reaction back into the enzyme catalytic functions. And we archive these different metabolic pathways to characterize which kind of reaction a specific substrate may get involved into. So I'm listing the CYP3A4 and 2B6 as an example. And that philosophy really expands to many other problems that we deal with. In here, the radar plot shows some of the representative ADME models we have in here, with the green being classification models, evaluated with AUC, and the blue being regression models valued with Spearman. I also want to point out that in the permeability predictions, we're also adding cyclic peptides because this modalities becoming more and more crucial. So we don't want to restrain ourselves with just the small molecules. And this also applies to antibody, as we'll talk about in a second. So now let's take a step back and consider what we've been talking about as the ineffectiveness of conventional small molecule design, which is we need to optimize one property one at a time. If we're looking at this problem in a higher level, then this is a multi-objective optimization problem. We need to take care of the ADME, DMPK properties, as well as the biological activity and its off-target profile. In addition, we want to pay attention to its synthetic accessibility because we don't want the chemistry to be the bottleneck. So how do we deal with the biological activity? Our solution is free energy perturbation. Our in-house FEP program is able to deal with non-covalent and covalent inhibitors, and we've fully deployed all of the modules onto our website. So let's take a look. Before we take a look at the module, I want to show you more statistics in here. In our recent publication, we benchmarked on 8 different systems, and it achieves a mean unsigned error of 0.68. kcal per mole. So that means it's sufficient to guide experimental design and reduce the total number of compounds to be synthesized. And then let's take a look at the website. We've built the module in a way where it's very smooth and easy to use, and the user wouldn't need to input a lot of, yeah, can you go into the non-covalent part? Yeah. So the user do not need to upload a ton of information. All they need to do is to upload a complex structure, PDB file, and a Mo2 file that consists a series of congeneric ligands. And the program is able to align and dock and determine the binding pose automatically. We would also build the perturbation map automatically for the users. We do offer a couple of other options, but the general guideline is to make sure that the parameters are as simple and as possible so that to alleviate the users the burden of choosing the parameters themselves. Because we have full access to the FEP module, we're able to expand its capability to covalent binders by incorporating quantum mechanics. The only difference between these two is we do need to assign the covalent residue. We could either upload the structure and point at the residue in this display window, and the corresponding residue number will pop up in the box. Or we could do it in the other way around by inputting the numbers in the box to be more specific. And at the same time, we're also developing a more automatic way of determining which of the residues is more likely to be reactive. And that could provide additional information for the users. All of these FEP calculations are by default using enhanced MD sampling. So in order to understand the MD trajectories, we deployed this interactive interface for the users to be able to view these different centroids by their overlays. So these are the representative structures of the top 3 clusters to see the subtle differences in the switch to region. We could also calculate RMSD, RMSF, and other relevant features on the fly so that to better review these structures. So now we have the second piece of the puzzle in here, which is the biological activity. And I believe the covalent FEP can partially resolve the off-target profile issue, although we do need additional tools to deal with that. For synthetic accessibility, we have rich experience in the chemistry field by offering not only our medicinal chemists, but also reaction database, and especially important with the negative data points as well as scarce reaction types. So we would be able to replenish the reaction data points in order to make the synthetic accessibility prediction more accurate. So this function's already been implemented in our small molecule de novo design program, which means the molecule will be automatically checked by this scores before being generated. In future, our vision is really to internalize also the ADMET and bioactivity prediction within the de novo design program, so that when we're generating these molecules, they are much more specific and much more efficient. And this is how we envision the roadmap of small molecule de novo design should look like. Now let's take a look at the in silico antibody design workflow. There are many different ways to form this entire workflow. And I'm showing the one that starts with just the antigen sequence. So this is the most challenging yet most intriguing problem. In that case, we will start from epitope mapping and followed by de novo designed sequences and structures, then affinity optimization and the developability index optimization. There could be other metrics that we keep monitoring during the process. And such a method is applicable to various different antibody formats, as listed on the right-hand side. What's really, really important in here is to bring in this antibody-specific large language models that's trained on over 50 million sequences and 10K complex structures. That can help us extract features, run epitope mapping, and keep updating these information as we generate these antibody sequences and structures. So in here, we're showing a very schematic workflow of the antibody workflow. So it's of the antibody design workflow. It's very similar to what I mentioned earlier in the case of PROTAC, because we're dealing with protein-protein interactions and we use AI-based scoring functions to help us rank order and filter these complex structures. So if we can move on to the antibody design interface, Junhao. This is slightly different than the MD trajectory analysis that happens on the fly. This does take a while, so we're showing you the preloaded project in here. So this is a membrane protein, and we identify these epitopes as highlighted in a heat map. And we choose the second patch in here to start our de novo design process. And in here, we're showing these design sequences and split their sequence display by their CDR regions. Upon clicking these buttons, we're able to view the structures on the fly, and tiny little mutations in here would impact the structure in the CDR region. And that's very crucial in designing, as a first step of designing. So followed by the de novo design step, we could either continue with antibody maturation, or the maturation step could be a standalone problem if we have any control antibodies to start with. So in this particular case, we're using a different example by loading the complex structure directly. So these mutant sequences are displayed in a way where we can easily view their differences in comparison with the wild type in the head. Since the mutations mostly happen in CDR regions, we do offer the automatic sequence logo plot for the research scientists to view the landscape of these mutations in these 6 regions. On top of the affinity that we usually care most, there are a series of developability index that matter a lot. And we are showing some of these in this table. This is not a complete list because the quantities that we care about vary depending on the problems we are working at. So if we can switch back to the slides where I'll show 2 more examples in terms of the affinity maturation and the developability index optimization. So for antibody design, as I mentioned earlier, it's not always increasing the affinity. It could be detuning the affinity or widening the gap of an antibody-antigen interaction under different conditions, or closing the gap of a same antibody against two different antigens. So that's the case of, that's the left-hand case. We start from an antigen that binds more closely, antibody that binds more closely with antigen B, but very loosely with antigen A. So our goal was to increase the affinity against antigen A and closing the gap between the two. So upon mutating 4 and 5 residues, we're able to reduce the affinity gap, and that's validated by SPR. In the second case, we're able to run pattern circumvention by mutating up to 14 different residues in the CDR region while still maintaining the affinity. And that's validated by not only ELISA and SPR, but also functional assay. And lastly, I want to show you the antibody developability and late-stage optimization that we've been really put a lot of efforts to. So on top, I'm showing 2 of the commonly used scoring functions. On the left-hand side, we're able to predict the BSA that corresponds to non-specific binding. And on the right-hand side, it's the aggregation score prediction of the FDA-approved antibodies. So in here, it's more like a profile of the entire antibody space, but in reality, we would use the score very explicitly in determining how risky they are, whether they are aggregation prone. Some of the key information I want to highlight in here is that while we have these base models that were trained on our very experienced experts, experimental data, we do want to take one step further, which is to build this transfer model to the clinical stage. So, for example, if we have the expression potential as a base model for HEK, we could use transfer learning to get the expression potential of CHL. And that same applies to in vitro PK or toxicity by building a mouse model as the base model for PK and toxicity and bring it to the clinical one that functions for human being. So those are what we've been dedicated at Viva for the past several years. And we believe we're responsible to bring the best of AI and show the rest of the world what AI can bring us and how far it can take us. Thank you very much.
Thank you so much, Dr. Qian. Dear guests, so that brings today's Enchantment of Drug Discovery launch event to a close. On behalf of Viva, thank you so much for being here, and your support means everything to us. So stay with us as we continue our journey for redefining the future of drug discovery. Thank you.